Hello, everyone. Good afternoon. My name is Isaiah Arkin. I'm the Dean of the College of Biomedicine here at CityU, and today it's my tremendous pleasure to introduce our speaker, Professor Muming Poo from the Neuroscience Chinese Academy of Sciences and Director of the Shanghai Center for Brain Science and Brain-Inspired Technology. When you have so many achievements as Professor Poo does, you need a page as big as this to read them all. But don't worry, I'm not going to do a complete justice. But just briefly, going through his esteemed career, he started off getting his undergraduate degree at Tsinghua University in Taiwan. Later on, went to Johns Hopkins to get his PhD. And then, as a faculty member, he moved from several of the most prestigious U.S. universities, including UC Irvine, Yale, Columbia, UCSD, and UC Berkeley, where he spent the longest period of time. He returned to China, to mainland, to head the Institute of Brain Sciences in Shanghai. And today he's going to tell us some of the exciting work that he has done about brain science and brain-inspired AI. Please, Professor Poo.
Thank you, Dean Arkin, for your introduction, and thank you for attending this lecture. I hope to present a lecture that everybody could understand, no matter which field you are in. That's my goal. It's a very high goal. So you can see whether I can accomplish that. So I'm talking about two science. One's the brain science, neuroscience. The other is AI. The intersection of these two areas is AI that inspired by brain science. So this is my rationale putting this together. Now, in about more than 100 years ago, in 1906, Nobel Prize for Physiology and Medicine was granted to two biologists, one Italian, Camillo Golgi, and one Spanish, Cajal, San Diego Cajal. They were awarded for the discovery of neural networks in the brain. This was a lucky strike by a special stain developed by Golgi. Now it's called Golgi stain. He happened to find a particular silver solution that can stain the brain section, stain the neuron sparsely. Only a few % of cells are stained. So revealing this complex web of processes in the brain. So if you had a very good staining, that stain all cells, you see everything black. So this was a lucky strike. Now this was the year that the Nobel Prize for Physics was awarded to J.J. Thomson for the discovery of the electron. It's very amazing events happening that we were told in that Nobel Prize lecture where Golgi stand up, attack basically the theory of Cajal. Probably Thomas knows about this story. And this is the only case that two awardees actually didn't agree completely with each other. The point is that Golgi feels that this web of network are continuum. Everything, there's no cells. Everything is linked together as a web. And while the Cajal think that this is still cells there, it's the cell process that making the link. But the cell has a gap. And he found that there are terminals of the axons from one cell that barely reach the cell body of the other cell. So there's a gap. So there's a contact point made between cells. So Cajal proposed the theory of neurons. Neurons, the cells in the brain. Now this is he proposed that it's a polarized structure with many dendrites and one single axon. Now you can see Cajal is an artist. He is actually likes photography and painting. He went into a wrong profession of medicine forced by his father. But he turns out he immediately finds his love in medicine, looking at the brain slices, showing the beautiful structure of the brain. So he spent all his life painting or drawing brain sections. I mean, he is the founder of the modern neurobiology. He actually made all the drawings. This is the cortex drawings. Now, to draw this structure from the structure shown here, it's not easy. You have to have a perception. You have to imagine what is it should look like, where the connection should be. So here you can see that he drew a cell with many dendrites and one single axon he could trace over long distances. This is what he finds as a polarized structure. And he also, in his drawings of hippocampus, he drew all these connections. He also drew errors. He said, these are static pictures, but he said, well, how does the information go? Maybe they go from the axon to the dendrite, and the dendrite seems to be a receiver of the information, and so he has errors in his graph. All these errors turn out to be correct. That's how information flows in hippocampus. We use HKIAS pictures even today. It's a very perception, he has a lot of insight into this system. So, definition. Now, this is HKIAS in his kitchen. He was a lecturer in the anatomy department of medical school in, I think, Barcelona. He spent his first month of salary, bought a monocular microscope on partial payment, actually. He has to started his own laboratory. In his kitchen, he set up a shelf, and he does all this standing and drawing. So in HKIAS view and in our view now today, the neuron is very simple. It is a polarized structure with dendrites, a lot of tree-like receiving end for the inputs, and a long, very long axons that sends information to the other cells and making synapse. Synapse is where the context made, where the information is transferred from one cell to another. Also, this is structure. Now, to know how the signals goes along in this network, it takes another 20, 30 years. The Edgar Adrian was the first one, making electrode, putting near the nerve fiber, detecting pulses of electrical current when you press the muscle. And you can see that these spikes, they all of similar amplitude, but with different frequency. He found that when you have a larger stimulus, higher load on the muscle, you get higher frequency, low frequency. So he decided that the signal was transmitted by these spikes, these impulses. Each is, the code is on the frequency of the spikes, not on the amplitude of spike. Every spike has similar amplitude. This is all known phenomenon of the neural signaling. And the timing and the frequency is the code for the signals across in the nerve. Then how does this action potential works? Now, it took Alan Hoskin and Andrew Huxley before and after the Second World War to figure out that these neurons, when they were able to get electron into the cell, he found the cell is actually negative by minus 60 millivolts inside. And when the stimulus comes, or you apply electrical current to depolarize the cell would burst into across certain threshold, would initiate a full-blown action potential of similar amplitude, 100 millivolts for 1 millisecond. Now that's the unit. That was the spike that Adrian detected extracellularly. This is intracellular recording, and they figure out that this is caused by particular flow of ions. Sodium ion flows in, cause this upshoot, and potassium ions flow out, cause this downward stroke. So this is transmission of action potential. Another 20 or 30 years, people figure out how the transmission gets from one cell to another through the release of a transmitter that activates the next cell. It's not the electrical current that goes straight across the gap. It's the chemical mediated transmission. Eccles and Katz figure this out. So this is all the all Nobel Prize winning work throughout the years. Major Nobel Prizes. Let me have a cartoon showing the process. The blood, the green vessels, that's the blood vessel. The action potential flow in the axon. It's the electrical signal is carried by ions. waves of ion flow in and out of the membrane, not by electron, unlike the cable transmission. Positive ion flows in, causes a depolarization that pushes the wave, the electrical wave forward until they reach the end. At the end of the nerve terminal, the axon terminal, there are packages of transmitter molecules that will be triggered to fuse with the membrane, release small molecules. These molecules turn on pores in the next cells. Let positive ion flow in, is depolarize the next cell. That if the pores is let only negative ion flow in, chloride ion flow in, it hyperpolarize the inhibitor cell. So you convert a positive signal at the end through either excitatory transmitter or inhibitor transmitter, you convert the sign to positive or negative sign. So this is how the synaptic transmission works. So now how we do look at the structure as a whole, brain as a whole. This is the human brain, about 1.5 kilogram. You can look at the structure by macroscopic brain imaging. You go to the hospital, take a MRI, magnetic resonance imaging, diffusion imaging, structural imaging at the millimeter resolutions will reveal bundles of nerve fibers in your brain. Now, if you have injury, you can quickly detect it. Now, each of this bundle revealed by diffusion MRI consists of thousands of axons. Right? It goes both directions, different cell types, so it's very hard to figure out how they really transmit, process information. But structurally, they are easy to detect at this high level, at this macroscopic level. If you really want to know the organization principle, you have to find out each different type of cell and their processes, their axons and dendrites, how they make connections. And this type of structural analysis we call mesoscopic, with micrometer resolution. A cell is about 10 microns, so micron resolution is required. This is all usually done by optical microscopy. You can also go deeper into nanometer scale using electron microscopy, where we have a lot of very complicated structures to analyze. There's too much information and too difficult to analyze, so people still refrain to study EM structure of the brain. A tiny piece of brain, it would give you, petabytes information that you hardly can handle. So we are, most of the neuroscientists still restricted figuring out at the cellular level how the connectivity is done. It's called mesoscopic connectome. Now, there are several problems to study. First, how many cells are there? It's amazing that after this early work of Cahal, more than 100 years ago, he started to distinguish different types of cells by morphology. He characterized hundreds of different types. We still don't know how many cell types are there in the brain. In the mouse, monkey, in the humans, the cell types in various type of various brain regions are being characterized by a technique called transcriptome analysis. Just you analyze the gene expression patterns because different genes are expressed at different levels in different cells. When they have a different expression pattern, they produce different proteins that cell will behave differently. So cell type can be defined by gene expression pattern, and that's called transcriptome analysis. You can do it in a single cell. And this technique is actually only 10 years ago, about 10 years old. And now it's very popular. In all tissues, people are doing single cell analysis. But in brain, we also want to know where that cell is located. So we call spatial transcriptome. We want to know at cellular resolution, the expression of different cells, the expression pattern across the entire brain. So this is a spatial transcriptome. Once we identify these cell types with particular gene expression, we have to identify how those genes are highly expressed, or what regulates them, to find the regulatory elements. Once you have the regulatory element, which is located usually in an open region of the chromatin, then you can use the elements regulatory element to express other things, exogenous protein that you can introduce, probes, you can introduce to specific cells, you can manipulate cells. So this is very important. Once you have that, you can put fluorescent probes, fluorescent protein expressed in a specific cell type, and then look at structures of single exon, including large, long-range projections, the input-output connections of each neuron type in each brain regions, using the cell type specific expression. Now, you also want to know the local circuits are a very intricate, complex web of terminals and cell bodies and many other non-neuronal cells. This has to be analyzed at EM levels, local circuits. Finally, there's a mapping of the function. Once you know structure, then you want to know how they work, how electrical signal are carried out. How do they code the electrical signal? Like, you know, this is a different cell type. Do they fire? What's their firing pattern? What do they do in a different brain functional state? So this is the 3 levels of brain imaging, and this is, I think, the current major task for the neuroscience. It's like the people started the Human Genome Project in the late 90s, last century. They want to know the structure of the genome, of the DNA. The sequencing technology is there, so they said, well, let's look at entire sequence of a human genome. And it took about 10 years to accomplish that. Here we look at the brain. We say, well, there are so many cell types there. We want to know the entire structure of this network, and that would be a human genome equivalent. It's called the connectome project in the brain connectomes. And at a single cell level, once you have the structure information, then you can start the function. So there's a lot of arguments in the field. People say, well, this is a boring work. I mean, it's detailed works, it takes a lot of analysis. There's no science in it, right? It's just like the sequencing of human genome. You have to be done. It has to be done. Once you know structure, then you can study gene regulation, how this gene interacts with other genes. Without the basic sequencing work, you cannot do the function. So there's a debate in China right now. Some people think this whole-scale, large-scale brain imaging is is too costly and not quite scientific interesting. So the mapping now faces the interesting thing is that we need to know the human brain. That's the essential 1.5 kilogram tissue. The animals we can use, most people use mouse. You know, it's 4,000 times smaller. Mouse is so small. Their structure, they have some structure like a human brain, but mostly the organization must be very different. So studying a mouse, which has been the standard preparation for studying brain science, is actually not adequate. So we need to look at a primate. Chimpanzee is out of picture. It's an endangered species. You cannot work on, you cannot do invasive experiment on chimpanzees. You can't get brain to the recording all this. But the closest animal model is macaque, monkey, a macaque monkey, or marmoset. This is a small monkey. Now these are the closest. So this is about 20 times smaller. So the macaque is the target that we are now doing. Most of the work we are doing is on macaque. The increase in size also accompanied by diversity of cell types, presumably also complexity in their connectivity. Now we need a lot of people to work on this, and a lot of groups are using different type of technology. We have to integrate, find standardized data acquisition methods and data sharing in order to achieve this goal. I mean, the Human Genome Project is a major project for several big institutions. Right? This Human Brain Connectome Project is going to be much, much more, 10 times, maybe 100 times more effort needed. So we have just established an international consortium for primate brain mapping just last month in Shanghai. 25 countries more than 100 scientists joined this effort to work together to hopefully in 15 years we'll resolve the connectivity of the primate, of the non-human primates, and by 2050, 25 years from now, human brain. Right? This also requires new development of new technology. So let me report to you some of the progress. First of all, we can already characterize all these cell types in macaque, monkey cortex, and the subcortical structures. Now this, for example, we have just based on the RNA gene expression, right? Different cells express different patterns of RNA. Those who express similar patterns, we group together, assign them to be same class or same subtype. Right? So this, you can see that many of the cells, it's like a classification trees. You can see these two types are closer in their gene expression pattern. They are closer to closer to the other together in a bigger class level. They belong to the same class. So there are many classes, many subtypes. Now these are tentative subtypes because depending on how deep is your sequencing effort, you can see differences, larger, more and more differences if your sequencing is more thorough, right? Deeper sequencing. So tentatively, we can say, well, at this level, at present technology, we can set at least 200 different subtypes in the cortex of the monkey that we need to use that to distinguish their function. So we can also know their spatial distribution, right? By sectioning this monkey brain, a thin section, 10-micron section, put on the large chips which can grab the mRNAs from the sections. A very sophisticated technology by BGI in Shenzhen, which gives us highest resolution, single-cell resolution of the mRNA expressed in this in different cells. Right? This is also requires a lot of work. You can also dissociate these cells in adjacent sections. You can do suspension of cells, do single-cell analysis, and do clustering and cell typing, and you can match that with the spatial expressed gene to know that these cell types indeed distribute in different parts of the cortex. And you can also check with adjacent section by traditional immunostaining histology to look at whether the gene you find is really there. Right? This is extensive work of many laboratories, probably the largest collaboration in neuroscience in China, involving more than 10 laboratories, more than 100 people involved in this work. So this is the first, most complete primate cell typing and a spatial distribution in the brain, both in the cortex and subcortical structures. There are various types of genes characterized. Even in the cortex, you can see even different layers of the cortex have a different type of cells. You can say have a subtypes colored with differently. The different cortical region, the layer-specific cell types are all different. So different cortical regions are performing different function. They are all in layer structure, usually 6 layers. Each layer have different cells. So we now know what these cells are expressing, at least knowing their chemical composition. Now the question is there's connectivity. Now then we need to label a single cell, sparsely a few cells, and trace their projection across the whole brain. And this technology is called fluorescence sectioning microtomography technique. And this is a technique also developed here in China by Qinmin Low's laboratories 10, 15 years ago. This involves injection of a fluorescent dye into the brain, and then fix the brain and do optical sections of the whole brain, and then reconstruction, the images you collect at different sections. by tracing the axons, individual axons, fluorescent labeled axons. This is very painstaking, very elaborate work that you can do only semi-automatic way to trace each axon, how they course along the entire brain to produce to reach their targets. Now the resolution capable of being achieved at cell resolution is sub-micron, right? XYZ resolution is 0.3, 0.3 by 1 micron resolution. Here's the example of about 50 mouse cortical cells, their projection across the entire brain. Cell body is hardly visible, but you can see that most of the cell is in its axon, in its process. All these resources are used to make connections. And cell body is tiny in this graph. This is the mouse. Now we can do it on monkey. This is recently accomplished. The injection, also sparsely labeled prefrontal cortex, the executive region, brain region that controls a lot of executive functions and a lot of reasoning are done in this region. The single cell, there's only 6 cell body you can now visible. Well, you only plot is 6. And then you can look at the entire course of projections, find their targets, the arborization of the axon, branching at the target region to make contact with the neurons in that local region. Now this, my interest in coming out of this work is you compare mouse and monkey, both 200 neurons, 200 prefrontal cortex neurons. Mouse is 200 times smaller. We actually enlarge it. You look at this 200 mouse neurons. Let's see. 200 mouse neurons across the entire brain. This is 200 monkey neurons across the entire brain. You can see the difference, right? What's the difference? That's really surprising difference. You thought in a monkey with much bigger brain, you should have more complicated projections. Actually not. The mouse projections are much more complicated than monkey. Why is that? It turns out that monkey, each cell body sends a long axon to its target and very few branches. While mouse neurons can send many branches to many targets. The reason for this is that the evolution from rodent to primate involves a large proliferation of neurons. Many neurons are born at a rapid rate for a long time, so you get a lot more cells. But evolution doesn't make the neuron better. Neuron is still similar neuron with similar capacity. So you take a mouse neuron and a monkey neuron, you put it into a culture, they grow at axon. You couldn't tell the difference by eye because the capacity for sending process is limited. So in order to be able to cover a large area, which is 200 times the size of the mouse, they have to be very specific. They have to target very accurately. The result is that in the monkey, you have a dedicated, more specific targeting of each cell. Now, cells are distributed. There are many more cells, 300 order of magnitude more cells, but each cell are much more specialized as a result. And that's really says something about our large AI neural networks, where you have a whole connection. You have many units, but you make a lot of connectivity, but that's a very inefficient way of processing information. The brain has actually evolved to be much more specific. Cells are should be more dedicated to specific target. And also, I should say that this specific targeting is not evolutionary endowed. Evolution endowed the ability to form very few targets for long distance communication, but which target to be formed? It probably requires pruning, rearrangement during development. When the monkey was born, when they were small, you look at their branching pattern actually much more. And then as they grew, the brain gets bigger, they start to be much more specialized. So there's a learning process starting from a much more branching and to a selection, pruning, to reach the final very dedicated connectivity. There's also another interesting structure. Now here's the cortex. Here's the subcortical region. In between, there's a very strange structure like a screen. It's called a claustrum. And it's a screen-like structure, a flat structure, which raised a lot of interest in people, especially Francis Crick. You probably know Francis Crick, which is the most brilliant theoretical biologist of 20th century. He discovered the double helix and spent his early days on the coding of gene. Then by the 1970s, he decided the real mysteries is in the brain. So he spent the rest of his life working on the brain, thinking of the brain. The last thing he thought about is consciousness. How does the consciousness arrive? He looked at this structure of claustrum. He said, well, this sits in the right place. And it turns out the claustrum makes connections to nearly all cortical areas and subcortical structures. That's strange. That's why they should do this. They said, well, this is probably a hub of integrating information across the entire brain. That's where maybe the site of consciousness, right? So Francis was actually very, very into this idea. He wrote his last article on July 28, the day he died in the hospital in La Jolla. This paper was published a year later, in which he asked, what are the cell types and their distribution in the claustrum? And that was 20 years ago. Nobody knows what cell type they are. What are the intra-claustrum connectivity and input-output connectivity? You connect to everything. Is there any regulation? Is there any regularity? Is there an organization so that they can serve us this consciousness center? And they also ask, what are the effects if we selectively activate or blocking specific connections to the various aspects of consciousness? Right? So he asked these three questions 20 years ago. So we were able to answer the first two questions recently. We sectioned many sections of the claustrum of the monkey. We performed the clustering analysis of the cell types. We defined the cell type, the distribution within the claustrum, distinguish many cell types. Here, some of the cell types are unique in the primate, not in the mouse. In the end, we found that indeed there's not only connection from all the cortical areas. Claustrum make connections with nearly all the subcortical regions, but the connection is orderly. In other words, specific domains of neurons covers particular area of the cortex. There's a topographic correspondence between the neurons in the claustrum and those in the cortex and in subcortical structures. And even more interesting, functions, cortical and subcortical structures are doing different things, various functions. Function, for example, when you try to keep memory, the memory process involves subcortical structure called hippocampus. If you take hippocampus away, you cannot form memory. So it's a memory forming. But the hippocampus makes connection with entorhinal cortex. It's storage of information. First step is to the entorhinal cortex. So this subcortical structure in the cortex turned out to be handled by same group of specialized neurons in the claustrum. Right? Similar function, their functions similar, but at different stages of their information processing. Same thing is the motor system, motor cortex and putamen, which is the basal ganglia, subcortical structure. They also are linked to the same group of structures in the claustrum. There are more things to regularity, including the very regular intra-claustrum connectivity, which seems to be playing a very important role. So this is work that needs to be continued. So in any case, we have started to look at very complicated connectivities in monkey. which is very close to humans. And there's some very good progress recently in China. About 30 research institutes and laboratories collected together, worked together, published a group of papers in Cell and Neurons in 2 or 3 months ago. And this large group of papers it goes from rodent to monkey to human. The comparison of different cell types in different species. And they started the mapping of the cell type and also starting to look at the connectivity. And the cell types in gene expression pattern and the projection pattern, do they match each other? That's an important question to be resolved. Right, so based on this, we actually is one of the bases for promotion of this international consortium. And many of these papers actually was international collaboration. Jan Mulder was actually author of several papers, several of these papers from Karolinska. So then let's talk about real function. The structure, we don't know how that will lead. Maybe before 2035, we'll know very little functions. But we know some functions already over the years. One most important function is neuroplasticity. We know that all the sensory, motor, cognitive experience are accompanied by electrical activity. Immediate consequence of activity is changes in structure and function of the synapse and neurons. We most consider synapse because synapses, you see that the release of transmitters and reception of transmitters, a lot of machinery there are very plastic, very easy to manipulate. So synapses become a place which are very plastic. Information relay are nicely, very efficiently controlled by synaptic transmission. So synaptic plasticity is very important. But neurons itself also are plastic. How they transmit information, how they spike, what frequency they spike. That's also plastic. Anyway, once this plasticity occur, the learning memory changes and cognition behavior changes. The process of changing the plasticity is called learning. When we learn, what we are doing is actually we change the structure and function of the connectivity. And the result of this change is what we call memory. So learning is the process that creates memory. Memory stores the learning experience. So when you hear my talk today, your brains change, hopefully forever. But I'm sure by tomorrow you still remember what I talked about, some of what I talked today. That's memory, that's learning, because that has to be put in somewhere. That's in the neuron and the synapses, right? That the modification has occurred. Both processing the information resides in the neural circuits, right? This is a very interesting. This is unlike a computer, the same network that processes information actually stored information in the connections. Not a stored information in a storage area, right? This is very different from the computer. Now, one of the most important influential figure in this plasticity field is Hep, a Canadian psychologist, a physiologist, who said 80 years ago, he wrote a book, he said that we have neurons that fire other neurons in the brain. Now, how do they change the synapse? Well, maybe if their firing pattern correlate, they have fired similarly, maybe that's a condition to change their synapse. This is just a hypothesis. There's no evidence at the time that this is really the case. And so he said, correlated pre- and post-synaptic activity cause synaptic strengthening and stabilization, a hypothesis, 1949. Now, then some other people, like Gunther Stein, said, for symmetry's sake, if two cells didn't fire together, if this cell didn't fire the other cell because this cell was inhibited by other input, so there is no activity, the activity doesn't correlate. Maybe they should be destabilized, weakened, and eliminated. Then you have both strengthening and up and down regulation is more symmetric modulation. Right? So this is the idea called cells that fire together, wire together. This has been a dogma in the field for nearly half a century. Even today, textbook is still saying cells fire together. Those cells fire together will wire together. Right? This is actually the idea applied to artificial neural network by John Hopfield in 1982. In his promotion of proposal of Hopfield network, attractor network uses HAPS learning rule to modify the synapse in the network. This is the most influential paper that won him the Nobel Prize in Physics last year. Right? So it's a one neuroscience-inspired AI. Now, there's evidence that indeed there's a synaptic modification. Now, in the 70s and 80s, people found if you have a high-frequency stimulation to a synapse with a repetitive stimulation, the response in the postsynaptic cell, the action of the transmitters, in open up channels in the postsynaptic cell, that response is elevated persistently for a long time. Or if it's high frequency for 1 second. But if you spread it out into 100 seconds, 1 hertz, right? Same number of pulses, you get the opposite result. You get a depression, so LTD. So if you want to remember anything what I taught today, remember LTP and LTD. If you know this, you can impress your friends in biology. Long-term potentiation, long-term depression is how activity store memories in your brain. Right? Now you can see explained by HAPS postulate. HAPS says, well, you high-frequency stimulation, the postsynaptic cell receive synaptic potential that add up to reach the threshold and fire. So you have a pre and postsynaptic cell fire. If you have a low stimulation, low frequency, the synaptic potential produced in the postsynaptic cell decay before the next pulse comes in. So you cannot reach the threshold, so you don't fire. Right? So this is the idea of cells fire together, wire together. It's confirmed physiologically. Right? But this transmission is happening in a very strange structure called dendritic spine in the excitatory synapse. There are 2 types. The inhibitory synapse, the terminal relief, a transmitter that opens the pores in the postsynaptic cell allows chloride ion to flow in. So it hyperpolarizes the cell. So it inhibits the cell. Excitatory terminal receive transmitter opens pores that allow positive ion to flow in. So it excites postsynaptic cell. Now, all these receptors are located in excitatory synapse in a structure called dendritic spines, a very strange protrusion on the dendrites. It turns out that this is a very useful structure, that there's a reason to have a spines on dendrites, because they can control the information flow, because they can change shape. The current flow generated at the tip, how much they reach the dendrite, there's a decay, the leakage of the current, so these changing shapes would determine the synaptic weight. Right? More permanently change the synaptic weight of the synapse. So functional LTP and LTD is function, right? The functional potentiation and depression. Those LTP, if you persistent created at the synapse, it causes swelling of the dendrite spines, making this synaptic current flow more smoothly. You have a stronger contribution to the dendrite. If you have an LTD depression, this spine shrinks. So the current created meets a high resistant conducting packs to reach the dendrite. So their contribution is small. So there's a structural changes that accompany the functional changes in the brain. You can actually observe this in real life, in vivo, by the so-called 2-photon imaging. You can stain the cell neurons with fluorescent dyes, and you can label different pathways with fluorescence. You can see the presynaptic axons making contact with postsynaptic dendrites and making synapse. And you can look at the same region of the brain at different time and find located that same synapse to see the changes with time. So we can actually see long-term changes dynamics of the synaptic connectivity. That's, this in vivo image is very useful. So by this, you can say, well, when we create a long-term memory, such as field conditioning, this is a very robust memory created in the mice. So you have a mice, you give a sound, you produce a sound. Immediately after the sound, you shock the animal with the electrically, right? Continuous pairing, sound shock. After a while, the mouse would freeze immediately after he hear the sound, because he predicts the shock is coming. This is called conditioning. Right? This conditioning. Right. This conditioning effect is very robust, and it lasts for a long time. It's called long-term memory. So if you look at the auditory cortex, which processes the information, the auditory cortex of this mouse, during the time they created, the 3-day period, they were storing the long-term shock memory, this long-term field memory. The synapse undergo some modifications. Many synapses change. The most dramatic is that the pre- and post-synaptic elements are modified by adding new elements. So here is the new spine, dendritic spine, added to the original contact. Here is a new presynaptic bouton terminals. So there are two boutons contacting the same spine, making the transmission more efficient. So in the end, what we found here is that the number of synapses that created, new synapses formed after the field is created, is increased. There are a lot more new synapses formed by this adding new elements. Not just local modification of the efficacy of transmission. It's a local modification of weight. It's not a gross changes of connectivity. But there are some, since it's very dynamic, there's also some synapse that gets eliminated. The number of eliminated synapses doesn't change. Those form more frequently. But this is unlike that in the right after birth, how the network was formed in the beginning. In the human, it's clear that all the neurons are present after birth, right after birth, right? And they die throughout the age, throughout their life. But the connectivity was very sparse. And within the first 2 years or 3 years, you see a greatly increased network formed. Right? This is the most dramatic picture I've seen in neuroscience, probably last decades. Right? This, unlike what we think that we are born with our intelligence. No, we are born with the capacity of form network, but the network are not formed. And this network formation is right at the time we acquire all our capabilities. We have to acquire the ability to stand up, to walk, that motor system networks are not there. You have to learn to produce an efficient motor system that you can stand up without falling. Right? And initially there are a lot of connectivity, but many of connectivity are pruned away. Only those good ones are remained. So you have a formation that larger than pruning. So you would count the number of synapses. It increases during the early childhood to a peak during the adolescence period. And then they decline with age. We're probably at this region going down. The number are going down, but our capability didn't really go down that much. As we'll see, the adult, we have shown that adult mice can learn field conditioning. Human can learn new language even in adult, even though not as efficiently. But this is plasticity, is ongoing plasticity is throughout life. But the early plasticity involves extensive rewiring, pruning and formation of synapse. So if this thing goes wrong, you have autism and schizophrenia, and they are known to be not enough pruning for autism, too much pruning for schizophrenia. So developmental and adult plasticity are different. There was large-scale rewiring of neural circuits in the developing brain. Right? This rewiring means they are formed at the beginning without precision, then they tune it to be more precise by use. The plasticity of the adult brain can also do learning and memory, but it involves much limited wiring. It's only the local remodeling of the synapse. Injury, on the other hand, can induce large wiring because you want to compensate the circuit. Even though the circuit formed after injury in the adult are not precise, not as precise. You don't go through the early developmental process of pruning and readjustment. If you have a new connection, new wiring formed in the injured adult brain, they don't have a good environment to allow you to form the original circuit. So the compensation is always limited. Right? So, but there's also idea in the clinical field that the developmental plasticity didn't really go away. They just buried. Right? They buried by the formation of very tight intercellular matrix called perineuronal net that surround the neurons. If you loosen those by enzymatic treatment, they're actually exhibiting much more plasticity, rewiring. This is an experiment done in animals. It inspires hopes that maybe in the adult brain we can create adult developmental plasticity by chemical treatment, by changing the excitatory inhibition balance and other things. So now HEPS, I think I'm almost short of time. Okay, let me see, HEPS cell assembly hypothesis. I think this is very important for the process, even though the idea was accumulated over the decades. I'm just summarizing here that the HEPS, the biggest contribution is not only his synaptic learning rule, but the cell assembly hypothesis. What he says that correlated activity strengthens the connectivity of activated neuron. But the result is forming an assembly of cells that store a particular memory. That's the key idea. I say, how is our perceptual memory stored? It's stored by modifying the synapse. We can link a group of cells together into assembly, a club that responsible for storing the memory. Memory is stored in this connectivity. This is his idea. Now, here's the example of the idea. Suppose we see a circle in our fields repetitively. You would activate a group of neurons in our brain. Each neuron are responding to one segment, curved segments of the sphere. But they are simultaneously activated, so their connectivity is strengthened. So you form a assembly for the memory of the circle. So once you have this assembly form, a few partial activation of this assembly, you would reactivate the entire assembly. That's the recall. Right? So you can have a partial recall. You see this, you know it's a circle. Right? So this is this idea. This idea can be generalized. You remember your grandmother's face. Well, each part of your grandmother's face actually are coded by a group of assembly of cells that codes for eye, for his nose, for his mouth. And all these are activated together. All these different components of your face are activated together in front of the grandmother's face, forming a super assembly of grandmother's face. This is assembly of mouth, nose, all this together. Now, once you have this super assembly of your grandmother's face, activation of your grandmother's part, you record grandmother's face. Right? So this is assembly. Now you can even extend the concept. Now grandmother is a concept. We remember her face as part of the concept. We also remember anything associated with her. Right? So by seeing her name, I recall everything about her. Right? That's a concept. Now you probably know this idea of an experiment in UCLA. Neurosurgeons recorded from a hippocampus of a patient and found that a particular neuron, single unit recording, that responds actively when the patient sees Jennifer Aniston, the famous actress. When Jennifer Aniston's picture appears, the cell fires. Any other movie star doesn't fire. But Jennifer Aniston's boyfriend's picture also trigger this neuron. And Jennifer Aniston, the name of Jennifer Aniston, also triggered the neuron to fire. So one just said, wow, we found a neuron that called the concept of Jennifer Aniston. And they call it concept neuron. Well, in fact, that neuron is a part of a huge assembly that links, is one of the cells, among millions of cells, that codes for Jennifer Aniston's face. Right? They are all linked to the Jennifer Aniston concept. So here's the hypothesis. The representation of a concept are assemblies that locate in various brain regions that are activated by various components of the concepts. Right? When the formation of the concepts is synchronized firing of the assembly, like your grandmother told in the story, and all the input about her converge to synchronize fire this assembly that represent different components. And the long-range connection among these located at different brain regions are linked into a concept. Right? So her name is probably in language-related areas. Her voice is probably in auditory region. Her face is probably in the visual face recognition area. So anyway, so how to the concept can be retrieved by activating a part. It's the same idea. So there's evidence on actually if you allow a patient with injected with radioactive glucose and sit in the PET imaging, which emit positrons, which is where more active uptake of glucose, radioactive glucose occurs. Give a visual stimulus, visual areas are lighted up. Listen, put some word, let the person subject listen to some words. Auditory region, auditory cortex are light up. You ask the patient to speak a few words. The vocal or the broadcast area, the language areas are activated. So now he said, close your eye, just think about the words that you just spoke. What does it mean? What's the meaning? You're thinking about the words, the same words. Now you see the entire brain lights up, nearly entire brain, many places light up. It says that simple thinking involves a concept. It's actually involves large area of the brain, different long range that are linked together in long-range connections. Now, this is the basis for the previous hypothesis I told you. There's about 25 years, or maybe almost 25 years ago, there's a twist in this story of HAPS learning rule. I found out we said HAPS says cells fire together, wire together. So we were asking how close they have to be fired together in order to wire together. If they just miss a little bit, right, or 1 second difference or 10 millisecond difference, do they still consider fire together? It turns out that the order of firing of presynaptic neuron and postsynaptic neuron is most critical. Right? So here is the input that sends the input before 10 milliseconds before postsynaptic cell was fired action potential. So you have pre-post firing. So presynaptic firing contribute a little bit to the postsynaptic firing. And then you get LTP. If this input fires after the postsynaptic cell already fired due to the other input, they are actually punished by depression. So both are 10 milliseconds apart. Right? These two neurons are fired 10 milliseconds apart. This before that potentiation. That before post before pre-depression. So the same 10 millisecond difference. That's why timing is more important than correlation. So this is the idea that was pushed by many laboratories, including Malcolm, Sagman, Bell, DeBell, and our laboratories. This is called correlated, temporary specific HAPS learning rule. Correlated pre- and post-synaptic activity can either strengthen or weaken the synapse, depending on the temporal order of the spiking. Pre before post, strengthening, post before weakening. This is called spike timing dependent plasticity it is found in all synapses in the brain, although there's variation of the time window. This is called, we can consider this a revised form of Hebb's learning rule. And this learning rule is now used in AI, a spiking neural network, very often, as a way to modify the synapse based on spiking timing. Right? If you consider timing is important in spiking neural network, this is the rule to apply. Well, I guess we can just go as long as a few more, 10 minutes? Okay, if you want to go, you can go. Right. I just want to play some game with you, I hear, just for fun. So here's let's say play a tune. Name that tune. Huh? Doesn't work. Anyway, okay, I just saved that step. I played 2 tunes, piano tunes, synthesized by computer. The first tune, you couldn't recognize it. The second tune, you immediately recognize is Beethoven's for Elise. I don't know, let's forget it. Anyway, I explain. The same tune, these 2 tunes, one you can recognize, the other you cannot, are the same scores, but one played backwards. Right? If you play backwards, you couldn't recognize the tune. So the memory is temporally specific. The sequence of events, an interval between events, is critical. Right? So that's why the time-independent plasticity is important. So here's the example. We have a cell assembly here, right? So we take the assembly to begin with, they are all uniformly connected. No strengthening, no weakening. Now we stretch it out into a stream, right? We give it a label. Then we give a sequence excitation. We started exciting this first, and then again this first, and to the end, and starting again. Always sequential excitation of this. Right? These thought experiments show that if you do this, then the connection, downstream connection, this cell to that cell, should be all strengthened, while the reverse connection should be all weakened because the pre-post idea, right? So once you have this, so cell assembly that store memories has stored a sequence memory, right? Like a tune, like a music theme. That has a sequence. If you reverse it, you don't have that memory. So once this memory is stored, then now if you hit only the beginning group of cells, because of strengthened activity, you produce you can recall the entire sequence. So this is how you recall a theme by playing a few notes, you can record the theme. That's the sequence memory. You can demonstrate experimentally. For example, you pose the eye detector, the brain, the visual system of a tadpole. By giving bars that sweep in 4 different directions. Right? They produce activity in a particular neurons. Right? The activity is usually similar before you have a training. Then if you give a training bar, which only sweep one direction, right, that the sequence excitation of that neurons. Then you come back with this testing, you'll find that the trained bars now create much bigger response. So this is a plasticity, a long-term plasticity, LTP-like plasticity that lasts for a long time. Now this plasticity is a sequence memory, and the interesting thing is that this memory can be erased if you allow the brain to have a spontaneous activity. So here's the bar that could produce this potentiation of the synapse in the brain. Now, if you have the cell that allows you to fire, then the memory is gone. It washes away. If the cell is not allowed to fire using voltage clamp methods, the memory can be kept. Right? So spontaneous activity in the brain, we usually don't know what they are doing. In fact, they play a role washing out useless information. The way we say that it's useless because if you give a spaced stimulus, then they can produce the spontaneous activity would be ineffective to wash out the memory. So here is the mass learning. You have 60 pulses to produce LTP. Right? If 60 pulses are applied at the same time, one shot, then the LTP, the potentiation drops, decays. Now, if you apply this 60 pulse by 20, 20, 20, spaced a few minutes apart, then even though the spontaneous activity is there, there is a potentiation. Now, how do we explain this? There's an optimum episode episode interval. So you have a first stimulus that creates a memory. Now, after a while, you create another memory. Before the decay of the first memory, you re-strengthen that. You maintain this memory at a high level. There's a biochemical process, it turns out, that once you keep this going for a while, then the biochemical process kicks in to stabilize that memory. So this is the result that with the optimum intervals of repeated learning, you have a memory. This is a checkout for LTP production and also for this natural visual production. Now, this is a way in our educational system. The psychologist knows the spaced learning is always best. Now, you don't want to learn at one shot and forget about it for a long time, and then you will not remember. But if you do the same amount of time of learning, but do a packaged interval learning, then you have a longer memory. So spaced learning is the important thing is that the memory has an optimum interval, right? The optimum interval is critical. If you have the interval is too long, the accumulation effects disappear. So for every memory, every type of memory, there's probably an optimal interval for converting them from a short-term into a long-term memory. This is what we learn. So I'm going to the last few slides into the AI. You all know the backpropagation algorithm. This is the basis of deep learning. Jeff Hinton, who invented this in 1986, got a Nobel Prize last year in physics, one of the most important contributions. And this very simple three-layer connectivity, the idea is that how do you change the network by learning? You change the network by teaching. You have supervised teachers supervising this output. If the output is correct from a particular unit, you don't change anything. If the output is incorrect, you change the input. Right? You use the error to modify the weight. So it's called backward propagation. You do layer by layer from the first layer to the deeper layer. And then this is the example, the most amazing example of net talk. You can input a set of letters, a word, a cat, a house, and now the output is the sound, the vowels and consonant sound. Now, if the teacher checks this output, see if it's correct. If it's incorrect, they modify the input, right? So you start with a lot of learning set of input with different letters and words. In the beginning, the network does nothing, like a child learning to speak, it's unclear. Gradually, they start to speak something intelligent. And after they really learn, you can just put in novel words, they produce it correctly. This is a net talk. When this was shown in 1986, right after backpropagation algorithm in the neuroscience community, everybody was impressed. Maybe our brain works this way. But Francis Crick said, no, brain doesn't work this way. Because brain information goes from dendrite to axon. When you have an output, the idea in the output, it doesn't flow back to input. So you cannot have backpropagation in our brain. So it's unbiological, even though there's a great excitement in the field. Right? This is what he made this comment. So I remember reading this article at the late 80s, and this is a special comment. It says it's unphysiological, no antidromic propagation of signals. A few years ago, in my lab, we have this network in culture. It says there are cultures interconnected. Well, we said, well, maybe this is a time we check whether there's a signal comes from the output to input. Right? The way to do that is you create a LTP or LTD by firing AB. In AB sequence, you get LTP. BABA firing, get LTD. Now, once you get LTD, that's the synapse from A to B. Right? Now we can ask the question, that's output. Would the input to A, which comes from the C, also changed on this A? That's upstream synapse. Or it's downstream synapse to other cells, would it be changed? So this can check whether Francis Crick's suspicion that this spread of synaptic modifications is unrealistic, unbiological. So here's 4 neurons making, in principle, 16 connections, 4 by 4, right? So there turns out that only opportunity for them to, by chance, only 9 connections have transmission, functional. So out of these nine connections between 4 neurons, we potentiate 1 neuron, 1 connections, E to 1, 2 to 1. And then we ask all the other connections, do they change? It turns out many connections are changed. Like this connection are potentiated. This is also potentiated compared to control. So there's a spread, and in fact, there's a backward spread. A lot of data show that when you create a LTP or LTD in the output of this neuron on this dendrite, if you create a LTP plus or minus, the signal goes backward to its input. That's called backpropagation, right? It can also propagate laterally to the collateral axon terminals of another cell. But interestingly, there's no forward propagation. So LTP, LTD created here doesn't spread to the output of the postsynaptic neuron. Right? So there's a rule. So the upshot of this, I should just quickly go to the end. It's shown in vivo, in the visual assistant. Is that that was a AI-inspired neuroscience, because it's a AI backpropagation algorithm inspired our experiment in neuroscience. So now we can go back. We say, well, we have a neuroscience finding. Does natural backpropagation of LTP, LTD in natural the synapse can help neural network, artificial neural network? So this experiment, this is our original backpropagation experiment, in collaboration with AI people in the Institute of Automation. We show that, in fact, if your teaching signal create LTP or LTD, by matching the output with your teaching signal, you can do a spike timing to create LTP and LTD, and then allow them to backpropagate to the synapse on the deeper layers. You actually help the efficiency of the spiking network. Now, without using BP algorithm. BP algorithm, as you know, is very costly in computation. And it actually, the error rate drops much faster. The energy, the computational cost is lower for 3 standard tests, standard benchmark tests. And also, another idea related to spike timing dependent plasticity is that there are many forms of this window. Some window are asymmetric, or some could be symmetric. This window are different property of the synapse. If you allow the synaptic modification to have a different type of SDTP, spike timing plasticity, you can also introduce a very interesting new properties. You differentiate different synapses. So the synapse that store information previously will not be easily erased by the future input. There's a problem in AI called the catastrophe of the network. The early memory stored will be erased by later memory in the sequence of learning. This was actually also reduced by having a brain introduced of a SDTP that are known to be variable. And it's known to be variable because we have found that in biological system there is there's a dopamine-dependent, neuromodulator-dependent modulation of the window of the spike timing. So the neuromodulation can work to produce so-called metaplasticity of the synapse. The synapse, not only they can change, the ability to change is called metaplasticity. The capacity to change can also be modified. And that, if you introduce that concept into the neural network, it actually helps to promote the ability of this network. So the property of brain network could be applied to artificial network by looking at the neural network structure. Now, we have a very efficient network in our brain that would endow in part by evolution. Evolution, while you are born, the evolution already produced somewhat loose connections that are different from lower animals, right? Because of gene mutation, everything associated with evolution. But after you are born, there's a huge amount of wiring process, and that eventually create the most efficient network. Now, basically, it's also actually a large data-driven modification. But eventually, we have a brain that only costs 30 watts to operate. Now, the training of AI is so costly because you started with the wiring that's no regularity, this full connection, and then you modify. You can have you change the parameters. Right? You can have billions of parameters to modify. You have billions of connectivity to do the learning. So you take a long time to learn, a lot of data to learn. But if you can use the network we found in the brain, in natural brain, the most efficient, the network structure that's already endowed, that may help to have a more efficient network. Also, the learning algorithm. How do you modify the weight? This is the learning algorithm. BP, the backpropagation is the most popular algorithm. But there could be other algorithms, as I point out. There are diversity of neuronal type. Connection patterns are different, can be forward, not only forward, but natural and backwards. There are multiple forms of synaptic neuronal plasticity. Also, memory has a different aspect. The consolidation process, short-term, long-term memory. The switch of a short-term memory into long-term memory has a rule, right? Spacing rule, for example. Spike timing is the importance, synchronous firing, binding of information through the HAPS assembly. Now, all these are properties that are not quite, were not applied. Now, we're not saying that the AI should simulate, it's a simulation of the brain. We are not doing simulation. We're using the principle of the brain science, hopefully that can help the design of the AI. And that's the potential benefit of having this one potential benefit. The other aspect of brain science and artificial intelligence, which I become aware of and thought a lot about in recent days, is the comparative study of human and artificial intelligence. We know that the artificial general intelligence is coming. Most people predict 5 to 10 years. Some people say 3 to 5 years. Now, this is intelligence that equal or surpass the human intelligence. But we don't know how to set a boundary for this. Especially when they were put into a physical system that can move around, a robot. Right? If they have that, you cannot easily turn it off. Right? That's a problem. Right? So we need to define what are various levels of intelligence of an artificial system. Right? Because without a good definition, we cannot really regulate or set a boundary for their development. So I'm proposing that we should use the method developed for studying the human intelligence to monitor quantitatively the emergence of various forms of human-like intelligence. We're not talking about their intelligence exactly the same as human, but they are very like the human. They are consciousness-like behavior. They are empathy-like behavior. All these human-like behaviors, we can monitor them. Right? Emotion, feeling, empathy, theory of mind, social behavior, cooperation, deceit, self-consciousness, metacognition, and purpose. Whether the AI system can have its own purpose, a free will, so-called. We also can learn from child development, child cognitive. We know that the wiring, during the wiring of the child's brain, the cognition of different level of cognition appeared with a sequence, very clearly as characterized by Piaget, a very stereotype development we can monitor. And also in child education process, how the child intelligence, how the child establishes character, its moral conduct. So and then design pre-training programs for the machine, for the acquisition of the world model. Every machine with intelligence, you have to train them to see the very complicated world, how do we react to the world. That's called world model. When we train them for the world model with this intelligent system, how do we endow them at the training process, a human-aligned ethics and moral values? That's a problem. And basically, we're saying that we need an educational system for the robots in order to have a robot that's a human-aligned and useful. Thank you.
Thank you very much, Professor Poo, for that wonderful and inspiring lecture. Questions from the audience? Please.
Thanks for the fascinating talk and the marvelous work. As far as I know, there is still a gap between the brain-inspired spike neural network with the traditional artificial neural network. The performance of the SNN is still comparably not as good as the back propagation-based models like transformer. Right. So I'm thinking that how do you identify this gap? And so nowadays, the neuromorphic computing is still has advantage in the energy saving scenario like the edge computing, but still we cannot benefit from its like how it can balance the flexibility and the catastrophic forgetting. So how do you identify the gap between the SNN and traditional network, and how can we solve that? Thank you.
I think it's not scientific but sociology, because there are very few people who want to spend a lot of time working on SNN and spiking neural network, because it takes time to break through all this develop efficient algorithm, as you say. While the deep learning algorithm is all there, they're so powerful so quickly. As long as you can apply them very rapidly, you get results much faster, you publish paper much faster. So that's the sociology that prevented the development of spiking neural network. But if you think about it, the timing of spiking, basically the back propagation is spike rates, right, as a way to modify to assay the weight of the synapse. But the timing of carry a lot of information, especially many of the information you want to process it's not static. Now the visual recognition, face recognition is static, But all the gesture, all the motion, any phenomena in the world are temporally coded structure. Right? So in principle, coding timing is very good. The spiking network is the best, most suitable network to work with. The reason that it's not developed as well is because it takes hard work. If you really want to work on it to overcome the barriers, right? In neuromorphic chips designs, in the end, it's also the architecture, right? What's the you have units that look like neurons, but how do they connect it together efficiently to perform the highly intelligent processing? That still, I think, requires the spiking networks offline machine learning algorithm that needs to be developed first before you implement it in the hardware.
Thank you. May I ask another question? Okay.
Please go ahead.
Thank you for a fascinating talk. You covered a long range. I want to ask you something that you talked about at the beginning of your talk, the comparison, the connectome of the macaque compared to the mouse. You said in the macaque, what you found was there was a more dedicated and specific targeting of the neurons in the macaque compared to the mouse. And so there's the larger volume brain, the smaller volume brain. There's a temptation to do scale, but let's just take the macaque and the mouse. What does that say with the idea of plasticity that you talked about quite extensively? You say that neuroplasticity is a structural and functional modification. And you might imagine in the mouse where there's not a specific targeting that there would be greater neuroplasticity, for example, if part of the brain was damaged, that the mouse might be able to accommodate more readily than the macaque where there are now specific areas. Are there any implications between macaque and mouse, or does the larger volume brain simply give you more complexity and diversity of the storage of memory?
Yes, that's an interesting issue that we have not addressed. How does this consequence of more dedicated targeting versus a lot of multi-targeting innovation by the mouse, how do they influence the plasticity aspect of the network? This is an interesting aspect we have not really addressed. In fact, I would guess that plasticity is found in both the monkey and in the mouse at a synapse level. It's the whether the need of targeting, whether the need of more dedicated targeting would affect the pruning process. I think that could be the contribution. In macaque, that's what different from mouse. The mouse doesn't have a pressure of resources. They can keep their multi-targeting the connectivity, but in the growth of a pyramidal axon, actually, for example, it goes a long way to our spinal cord. But they also have starting early have some branches in other brain areas. Those were pruned during development because they want to keep the most important targets. So that limitation may affect the plasticity of improving. Something needs to be studied, to be addressed specifically for this, because of this evolutionary constraint.
Should I stand up or?
Yeah,
Just,
please.
okay. Thank you for the fantastic talk. So, my research area is spiking neural network and also brain-inspired efficient large language models now. So I think it's a great point that you mentioned. I think ever since 2019, and spiking neural network, it just moves more towards just artificial neural networks, but just far away from the real brain inspiration. So I think maybe after today, I will just read more.
The hybrid system. it's a pseudo spiking network.
Exactly.
Yes.
We just basically use the leaf neuron, and then it's really close to the recurrent neural network, I think, was the current lesson. So maybe after today, I will just read more neuroscience papers and to see if I can get more principles.
There's a small community still working on it, at least in China, on the spiking neural network, how to improve the algorithm.
Okay, thank you. And for the large language model, I think one big problem that we face now is that it's really hard to integrate the different modalities data, say like audio or like context or videos. And that the grandma's example is really great, right? Because the grandma's name or face or smell, then you can all unite as concept. Do you think that there's some principle we can get from this concept integration, and then we can inspire the larger language models?
Yes, I think very important. So now the big language model, which is just analyze a sequence of symbols,
Yeah.
The text symbols. How do they integrate with a multisensory system? That's actually the biggest barrier I can see. The multisensory integration is the key for human interaction with an environment. One of the key aspect is the information from different channels, from vision, hearing, proprioceptive sense. All these senses are processed in a very strict time-dependent manner. They are correlated. So I see you talking. I see your sound coming from your mouth. Those are synchronized information. A channel has to be synchronized. So spiking network have to be basically the best chips I know to multi-sensory integration. Use the spiking network as a basis, because you can synchronize different channels. And then you integrate in our human brain. There's an interesting processing. It's not a different channel. You finally integrate it. The integration goes along the way at different levels. Vision and hearing are talking to each other at the superior clavicular level, at the cortical level, at the thalamus level. They all have a crosstalk. So there's integration depends on various levels of interactions, eventually to get your final perceptual the image. Yeah. So that could be a very interesting aspect to really incorporate into the AI system.
Okay, thank you.
Well, I think after an hour and a half, Professor Poo deserves a break. So please join me, everyone, in thanking him again for a wonderful seminar.
Thank you.